Hi, my name is Ameya Phadke. I'm a fourth-year PhD student in the Department of Bioengineering. My Socrates project was titled Stem Cells and Smart Materials, and I taught it at Mesa High School in partnership with Ryan Benedict, a biology teacher there. We taught in a 10th grade general biology classroom from the 2011 to 2012 academic year. So the objectives of this project primarily were, one, to introduce the idea of cells as very dynamic creatures, and two, to sort of bridge that with my research in stem cells and smart materials. And so this starts off by first introducing students to the fact that cells, like I said, are very dynamic creatures. They can sense their surroundings, they can move around, they can make protein in response to certain chemicals. Now, cells use a variety of different organelles to use this, so it's a very cell biology focused activity. But it also ties this with the central dogma that is, DNA, RNA to protein. Since typically a cell's response involves taking a signal and then that signal resulting in some protein being made or destroyed. Now, since cell biology and the central dogma are typically taught as very separate units, the idea of this activity was to bring them both together. Now, we do this using a lot of very fun and engaging methods, and I'll get into this a little later. The other objective is to introduce students to the concept of stem cells, where you have a population of cells in your body that can transform into very many tissues. And this is really how an entire organism is created from an embryo. And so it goes a little bit into that. It also uses that as a platform to go into the ethical considerations of stem cell and animal research. It really pushes students to think about these issues on their own and really gets them thinking about this. Again, introducing them to very cutting-edge research in a fun and engaging way. So in the classroom, this activity would start out first by showing the students a video, and this is a video of cells walking on the surface of a material. Now, as you can see here, the cells were moving. They weren't just very static creatures, which is the image that a lot of students have of stem cells, or cells in general. And so this really gets them thinking, well, okay, cells can move around. How do they do this? And then there's a little bit of an introductory PowerPoint on what kinds of components are involved in this process. Now, we simplified this a lot to include only the membrane receptor, the nucleus, the Golgi complex, the tRNAs, and the ribosome. However, you can make this as simple or as complicated as you like. It's really very open to modification. Now, the way we do this is the students will enact various organelles in the cell, and so we'll have some students do the membrane receptors. They'll pick up the signal and pass it to the student who plays the nucleus. The nucleus will transcribe the gene, pass it to the ribosome. The ribosome will then look at the transcribed gene, and it's basically a sequence of colors since our amino acids are different colored Legos. The ribosome will then ask the tRNAs to bring him or her the right amino acids, pass it on to the Golgi complex, and then the Golgi complex has to figure out where this protein goes, either inside the cell or outside the cell, based on what the first color of the protein is. And so it's a very teamwork associated activity where the students have to work as a team, go all the way from an external component or an external signal, all the way to a finished protein that's either secreted or kept inside the cell. And so it does a really nice job of bridging the various organelles in the cell with the central dogma. So, DNA turns to mRNA and then that's made into protein. So this is a really interesting way to look at it. Students have a lot of fun. The way we set it up was we typically had 2 or 3 teams of cells running against each other, and then those cells would make protein, and the first cell to make 2 finished proteins was the winner. and we had different prizes and different things that they could work for in that. However, the point being that a lot of students enjoyed this a lot, and it did a particularly good job of engaging students like athletes that typically weren't engaged in science. So anyway, moving forward, we then go into a unit on stem cells, and again, we talk about, some cutting-edge stem cell research. I talk about my own research in developing synthetic bone, and then we play a little bit of a stem cell board game. Now, as you can see on the board, students start out at the blastocyst stage and go all the way down through a series of moves till they become differentiated cells. Now, every student in the class does this, and at the end of the class, we have many different types of tissue represented in the classroom. And then just for fun, we take a little bit of a poll of how many cell types we have, and then we actually draw the creature that we would be if this was a real animal. And so once we got a creature that had a lot of eyes and no bones, we had one that had a lot of muscle and no eyes. So it's a little fun, but the students really get an idea of, okay, these are the things that stem cells have to go through to make a complete organism. And so it does a really cool job of really establishing that understanding. Then at the end, we do a little bit of Q&A so students can ask some questions that they might have about stem cell research. They can address some of the misconceptions that they have based on this, based on what's in the media. And so it does a really good job of pushing students to really think for themselves, and not everything that you hear in the media is strictly true. What I particularly like about this activity is it takes relatively complicated concepts like stem cell engineering and stuff like cell signaling and presents them in very fun and engaging ways. So for example, the teamwork game, students get really into that. I'm not kidding, we actually had to break up a couple of fights because students got too into the activity and started to fight with people from other cells. But point being, they really enjoyed it, and the comment cards reiterated this. So they said, I had a lot of fun, I didn't know that learning could be fun, and we got to play a game, but we still learned something. So I was really happy to see that. This activity, was developed with the input of a graduate student, myself, as well as a science teacher, Ryan Benedict, who had a lot of experience teaching classrooms. So it's a great way to bring there was a best of both worlds approach that really worked. Next, one thing that I particularly liked in addition to all of these is the fact that it's very easy to translate from person to person. You don't need any complicated equipment. You just need Legos, pieces of paper, sheet protectors, and you can make it as complicated or as simple as you like I said. So you can extend it to different applications. You can add lysosomes, you can add membrane, you can add all sorts of things into it. Regarding the stem cell game, again, it's a very fun and almost a very imaginative way of dealing with how stem cells actually function and giving students an idea of what they actually do. Students get to play with Play-Doh, so they always have fun there. And I really wanted to make the focus of my project, something that can be easily translated from person to person, and something that really can pull in students that typically would not be engaged in science. So for example, the competitive nature of the kinesthetic activity, or just the board game nature of the stem cells board game activity, I really wanted to engage them in a fun way, but still give them very substantial learning material and learn concepts that they might not have been able to grasp otherwise. Like I said, this does a really nice job of fitting in various different units together at the interface. And I wanted to do this since I, as a scientist, also work at the interface of chemistry, biology, and medicine. So all in all, I had a great time developing this project, and I hope that you have a great time implementing it in your classroom. Thank you.